All right, everybody, we're back again, continuing our adaptation unit. This time we're going to talk about adaptation in a little different way. We're going to talk about how human beings can actually manipulate something to adapt to benefit us. That's called bioengineering. And what I mean by that is, what if we could take a virus or a bacteria or a fungus or anything small, microscopic like, and change its DNA around a little bit or its RNA around a little bit to tell it what to do. To say, instead of eating our cells, you go eat bad cells that can cause us cancer, get rid of those and then self-destruct, or just shut down and just kind of float around in me until we dispose of you in some way. That's something that scientists are really thinking about right now, especially when it comes to diseases like cancers and heart disease and other things that are pretty pandemic around the world and cause a lot of people to suffer. So let's think about it in this way. Let's say we could take a virus. Now, viruses are controversial to begin with. Most people are deathly afraid of viruses. Viruses are things that cause bad things to happen to us. That's not really true. Most microscopic organisms, viruses and bacteria, are actually helpful to us. They do things to help us live better, live longer. There's bacteria in your stomach that allows your digestion to be more favorable to you. There are bacteria inside your eyelashes that eat other bacteria that could harm your eyes. There are viruses in you that allow other viruses to not harm you. They will go after those viruses. Now, sometimes when it comes to these bacteria and viruses, our own immune system will get rid of them. But because there's so many of them, usually there's enough to last and continue to help us, even though some of them might be taken out by our immune system, our white blood cells. It's very, very interesting to note that you have about 6 trillion cells in your body. Maybe might have a little bit more, because a little bigger. Some people might have a little less, but on average, 6 trillion cells. And you actually have more bacteria and viruses on you than you have cells. Maybe 3 times more, 4 times more, whatever it is, but there's more of that because they can sometimes be smaller than your cells and be on your cells. So you actually have a lot more foreign material in your body than you have your own material. It's kind of an interesting thing to note. Now, viruses are controversial also because some scientists don't really consider them to be a living thing. All right? To be living, you need to, interact with water, take in some sort of energy, sunlight or food to help you maintain. You got to grow and you got to reproduce and all these different things. Well, viruses don't really do that. First of all, most don't even have DNA. They have RNA. And also, they don't reproduce, they replicate. They become the same little thing over and over and over again. That's what makes them pesky and hard to combat. But at the same time, they're tough. They're strong, they're hardy. And that's why scientists look at viruses as a means to manipulate so that we can send it into your body to attack, say, like cancerous liver cells, and your body might try to fight back, but the virus might succeed regardless. So, in the order for you to understand this more, we're not going to talk about both DNA and RNA. That's just too difficult to talk about, especially with 3rd through 5th graders, 2nd graders, whatever you might have. We're just going to talk about DNA. And what we're going to mainly talk about is the code of DNA. The code of DNA is 4 nucleotide bases. There is A, which is adenine. There is T, which is thymine. There is C, which is cytosine. And there's G, which is guanine. They pair up in a certain way. A pairs to T, T pairs to A. C pairs to G, G pairs to C. That's it. Those 4 nucleotide bases create the DNA code that makes every single person on this planet, every single organism on this planet, other than some viruses, do, look, and behave the way they are. It's that 4-coded DNA. So what if you could take the code of a DNA and take certain sections out and add certain sections in to give it instructions to do something? So let's say viruses had DNA, or we could put DNA into a virus and tell it what to do. That's what bioengineering is. It's taking a living thing, using engineering principles, and making it do something that's beneficial to us. So let's think about this in the sense of, what if the kids could create their own virus? What if they could create a virus that could combat a certain ailment or disease? Let's take this one step at a time. First thing you're going to want to do is you're going to want to go ahead and give each of your students one of these index cards. This is going to help them do a couple of things. It's going to help them understand how nucleotide bases pair up. It's going to help them write down the sort of disease they would like to counteract. And it's going to help them change the DNA code that you give them into a new code that will make the DNA and the virus do what you want it to do. So let's think about this for a minute. What if you were to write an 8-section code down? ATCG code. Let's just go ahead and do that right now. I'm not going to think about it. I'm just going to write 8 letters and see what happens. All right. This is what I wrote down. I wrote down A, T, C, C, G, T, G, A. Now that's only one half of the code. All right? DNA is a base pair. That means with every base nucleotide base, it has another one next to it or across from it that would make it a pair. So remember, A is always with T, C is always with G. So if you were to complete this code on the bottom, if you had A, you would put a T underneath it. If you had T, you put an A. If you have C, you put a G. If you have G, you put a C, and so on. So you can go ahead and complete this base code, and there it is. So that's one little code and one strand of DNA. Now, your students are going to have a chance to make DNA. So you're going to want to explain DNA a little bit further than just the base codes. But we'll go step by step through that. First thing is DNA is a long chain molecule. It's known as a polymer. Okay? It's long chain, has repeating subunits, continues, continues, continues. Proteins are also polymers. All right? One DNA strand inside of the nucleus of one cell, if it was unraveled, taken out of that cell, taken out of that nucleus, and spread end to end, would be the size of me. I'm 6'8". It would be 2 inches shorter than me. That's how long these codes are. They could have billions and billions of nucleotide bases with different information. But we have a project called the Human Genome Project that has isolated the code of DNA. So we know what certain codes can do and what they mean. Now all we have to do is change it. So let's say you have a child and they want to make it very personal, the ailment they want to eradicate. Let's say they have an uncle who has lung cancer. So they write down on their card, virus will fight lung cancer. All right? So they have that written down. Now the next thing they're going to do is this. They're going to take the original code that you gave them and they're going to change it. All right? They're going to write a new code down the way they want to. All right? So, instead of being A, T, C, C, G, T, G, A, they're going to write their own code. And it can be simple, it can be complex, or it can be whatever they want. So in this case, I made a code that's A, T, G, C, G, A, T, T. Underneath it, you complete the base pair. You have T, A, C, G, C, T, A, A. That is their new code. And that's what's going to help their virus combat the lung cancer. Now, once you're done with that part, you're going to make a strand of DNA. What you might want to do is beforehand write down on the board what each of the straws that we give you stand for. So a yellow straw could be T, thymine. The orange straw can be A, adenine. The blue straw can be C, cytosine. And the pink straw can be G, guanine. These are the straws. little cocktail straws, okay? And they're going to use these straws, they're going to measure them out using this ruler, 1 inch apiece, and they're going to create their 8-stranded DNA code. Now, instead of me sitting here and making a DNA strand, which could take 10, 15, 20 minutes, I already made one beforehand, so you can see what it looks like. This right here is my DNA strand. As you can see, yellow and orange pair up because that's A and T, or T and A, and pink and blue pair up because that's C and G. All right? Now, the tape that puts them together, that's known as the hydrogen bond. And these two pieces of masking tape, one piece to stick them on, one piece to cover the other part up, that's the sugar phosphate backbone. Once you have this completed, you can make it do what DNA does, which is to twist and get smaller. All right? So you'll have them make this little strand of DNA, you'll have them make this card, and they're going to be placed somewhere. This is where they're going to be placed, inside this box. This Chinese takeaway box is a viral capsid. It's the body, the tough body of a virus. So what you're going to do is you're going to fold this up, put that in there. You're going to twist this up, put that in there, and you're going to close this up. Now we want to imagine this as a delivery system. You know FedEx. So instead of FedEx, we're going to call this Virex. Federal Express, Virus Express. You get the idea. Now, if we sent this capsid in here like this, all kinds of bad things are going to happen. It's going to go out of control. It's going to start to do things it shouldn't be doing. The white blood cells are going to attack it, maybe destroy it. So you have to also have proteins around this virus capsid, or stuck to this virus capsid, to give it information and instructions what to do. These right here, these stickers, are your proteins. So you can take one sticker and you can stick it onto your box, and you can write something like rush delivery. That way, the virus knows not to sit around. It knows to go right to where it needs to go to. And where does it need to go to? Well, we can take an address label, stick it on the top of the box, and this is lung cancer, so go to lungs. So just go to lungs. All right? So rush delivery, go to lungs, and then there's other proteins. You can take a green sticker, stick it on here. Tell the white blood cells to leave it alone. So I put supposed to be here. White blood cells do not attack. Simple enough. Take another sticker and put it on the side. Give us some more instructions. One thing you're going to want to take care of is make sure that the virus doesn't stay too long, doesn't live past its warranty, basically. Once it gets rid of all the cancerous cells in the lungs, you're going to tell it to either leave the body or self-destruct in some way so that it doesn't start to mutate and become something harmful to us. So when its job is done, its job is done. So on this one I put cancer cells eradicated, leave body. So it's somehow going to travel out of the body. I don't know, maybe it'll stand in the lungs and when you sneeze, out it'll go. Or maybe it'll just go down the digestive system and be digested, but whatever it's going to do, once it's done its job and it has to go. Okay? So these are the sort of things you can tell it what to do, these proteins. And by making this viral capsid, this DNA strand, this card with instructions, these different proteins, the kids have actually engineered something that would benefit humanity if it was really a virus. I know it's just a model, but it's getting the idea across that this is the future. And these are the things that scientists are going to be able to do someday. So good luck with this. This one is a little hard. You're going to have to help your kids out a little bit more. If you have little kids, I would really suggest on this one that you do it one all together, especially if you have kids like, kindergarten and below. But they still will understand what's going on. And this one also is a very good one to do journaling with, because they can put their thought process behind why they're building it a certain way and why they're doing what they're doing to make it what it is. Okay? So that's that one, and we will see you next time.